Hello, my name is Predrag Radivojac, and I'm presenting here our work titled The Field of Protein Function Prediction as Viewed by Different Domain Scientists. This is a joint work with Rashika Ramola and Professor Ido Friedberg, and as of now, you can find this work on bioRxiv. We as a group have a long-standing interest in protein function, in annotation of protein function, and in the prediction of protein function. In this work, we have identified three core communities that are associated with protein function research, and we decided to survey them to learn more about this field. These communities are experimental scientists, biocurators, and computational biologists. Experimental scientists, they conduct biological research, they test hypotheses, and they publish the results of their research in papers. Biocurators take these papers as a start, they curate this information, they systematize this information, they also develop ontologies, and then they store this information in biological knowledge bases. Computational biologists generally rely on the curated information from biocurators, and they develop methods for analysis of protein function and prediction of protein function. They also develop software that can be used by experimental scientists, for example, to further guide the studies of protein function. And therefore, given those three communities, we would like to survey them to assess the state of affairs in this field, to understand this field better. So our goal was not just to understand their views on this research area, but also some of their practices in this research area. We would like to assess the quality of the methods and usefulness of these methods for prediction of protein function to experimental scientists. We would like to understand the nature and level of interaction between these groups in this field of research. And throughout all of that, we would like to highlight positives, but also reveal the bottlenecks in the field. Our participants were experts in the field, and our rationale was that only experts in the field who work in the field can help us understand the field itself. And therefore, our requirement was that each participant had to have a PhD and publications in this area. We have recruited or contacted 169 scientists, of which 82 have finally participated in the survey. Of those, there's 22 experimental biologists from 12 different institutions, 19 biocurators from 10 institutions, and 41 computational biologists from 38 different institutions. Our survey was online, it was done via Qualtrics forms, and participants were offered $10 gift card for completing. So there's these three groups of scientists, and each of them had a general questionnaire. Those questionnaires were different for different groups, but every person within a group had the same questionnaire. So we asked about their experience, about their training, about their familiarity with databases and software in protein function research, and we also asked them a little bit about their own job. For experimental scientists, we also had a specific questionnaire, which is we gave them predictions from modern tools on protein function prediction, and we asked them to evaluate the quality of that prediction on proteins that they understand well. And then for experimental scientists, we also had a comparative questionnaire where we side by side showed them the predictions from the top algorithms, at least based on critical assessments, and the baseline algorithms, and then we asked them to compare the two. So this is the first result, and you can see here on the left that on years of experience, the blue bar is always experimentalists, the red bars are biocurators, and the yellow bars are computational biologists. And so this is a pretty experienced cohort, but predominantly more than 10 years of experience in the field. On the right-hand side, we see the fields of training, where experimentalists and biocurators are generally or predominantly trained in biology. All the biocurators have expertise in computer science, whereas computational biologists mostly have been trained in computer science, but they also have experience in biology or maybe statistics and some other areas such as physics. This is familiarity with databases and software. We asked everybody, we showed everybody a lot of different databases and several software packages, and we asked them to rate their familiarity with these databases or software. So 4 meant used frequently, and 0 meant that they were not familiar with these tools. Here we can see that for general databases, biocurators and computational biologists were pretty familiar and more familiar with these databases than experimentalists, and those were UniProt, SwissProt, and Gene Ontology. This does not hold for PDB, where experimentalists were actually quite as familiar as biocurators and computational biologists. Across many other databases, the general trend is that biocurators are the most familiar with databases, and this particularly holds for this part here, which are model organism databases. The reason for this, we believe, is that for model organism databases, it's part of the job for biocurators to understand multiple databases, but experimentalists will generally be interested in maybe only one of those, whereas computational biologists will be generally interested in general databases. And therefore, biocurators are the most familiar with all databases, followed by computational biologists and then experimental scientists. In terms of software, we show them several software packages, and this is only one result where we find that there's really no difference in their familiarity with BLAST between experimentalists, biocurators, and computational biologists. Therefore, all of these groups are quite familiar with the major databases in software in this field. Now, we ask experimentalists about their use of software for understanding protein function. Interestingly, 64% of those say they do not use any software to understand protein function, and 36% say that they do use some software. So when we asked them what they use, they said SciBlast, HHSpread, String, SwissProt, Interpro, and iTaser. Some of these are databases. They do list computational predictions, but others are just tools for maybe finding remote homologs and similar sequences in general. From this, we conclude that experimentalists do use various types of databases and tools, but they rarely use modern predictors for protein function prediction. Now, we gave them the outputs of protein function predictors of modern tools that we call here state-of-the-art methods, and some of them are actually new and others a little bit older. And so we randomly picked one, and then we made predictions for each experimental scientist on proteins that they're familiar with, and also two general proteins where we expected some general knowledge. And then we asked them to say whether this particular prediction is known, useful, surprising possible, surprising doubtful, wrong, or they can leave it unlabeled. So this is if they don't check any boxes. Here we find that most of the time they say that the term is known, which is understandable because many of these protein function prediction tools use those particular terms in their training set. But then what we are really interested in here is that quite a bit of time, for 700 terms roughly, for useful and surprising possible, they actually find those terms of interest for them. And this is both in molecular function, biological process, and cellular component from the gene ontology. Although these particular predictions were also accompanied with those that they find surprising and doubtful or plain wrong, we still feel that this is a very positive outcome, that even the experts in the field have found these predictions to be interesting for them. And then we asked them on the right-hand side a very simple question: did the algorithm do a good job? And we see that about 40% of them said yes, about 20% of them say no, and some of them have said something that we characterize as somewhat. This is because it was a textual field, and some of them were just unable to say if the algorithm did a good job. Now, finally, we asked experimental scientists, we showed them side-by-side predictions from state-of-the-art tools and baseline algorithms. And from the baseline methods, we used naive, top, blast, and gotcha algorithms, and you can see the reference on the right-hand side. So when we show them these predictions, we ask them which one is better. And we find that most of the time, they cannot distinguish between the baseline algorithm and state-of-the-art algorithm. In 25% of the cases, it was a tie, whereas the wins were randomly, certainly not randomly, but distributed between wins of the state-of-the-art and wins of the baseline. One reason for that could be that these differences between algorithms are very small. Another reason is that we only show them top 25 best predictions by each tool to limit the overload of information to experimental scientists. Now we move to ontologies. Here the question was to each of the core communities. On the left-hand side, how useful do you think is a Go annotation for an experimental scientist? And then on the right-hand side, we see how well do you think Go terms describe protein function? Here we see differences between these three communities. Experimentalists find Go annotation for themselves to be somewhat to moderately useful. Biocurators think it's very useful, that information for experimental scientists. And computational biologists are somewhere in between, going from not useful to very useful. We see statistically significant differences between these groups. But when we ask them how well do you think GoTerm described protein function, we see that, again, biocurators are the most optimistic. They think that they describe them very well, and a smaller number of them think well enough. Whereas computational biologists and experimentalists actually have very similar responses centered around well enough, and only a relatively small fraction of them said that GoTerms do not well describe protein function. Now we asked them, this is predominantly biocurators and computational biologists because they understand prediction better than experimental scientists. We asked them, what are the chief bottlenecks in protein function prediction? And we see that in terms of available data and ontologies, computational biologists find those to be the chief bottlenecks in the field, as opposed to biocurators. But then when we move to methodology and evaluation, we see more biocurators find this to be the chief bottleneck than computational biologists. Therefore, each group has a little bit more appreciation for their own field and think that the problem is perhaps in the other part of the subfield. And some of them also said other, and then we asked them what other problems are there. We saw very interesting responses such as explainability, advertising of the field, and popularization of the field, and generally educating people about each other's work. And then we asked them also about their interaction with experimental scientists. We see here that biocurators, 50% of them or more, have extensive relationship with experimental scientists, and then occasional and then minor, whereas computational biologists have a little bit of less interaction with experimental scientists, but the differences were really not statistically significant. To conclude, we surveyed 3 core communities to learn more about the field of protein function prediction, and we identify several common views in the field and also several idiosyncrasies for these existing scientists. For example, everybody is more appreciative of their own field, and they do not quite understand as much the nature of the field for the other group. We also identify that there are challenges in this field, especially between the different subgroups, but also there's reasons for optimism. For example, experimental scientists, although a challenge, they do not use computational software. If they were to use this software, it seems like they would find many of these predictions to be useful or surprising and possible, so they could perhaps follow up on their experiments. Biocurators have a large appreciation for ontologies, but it seems that that information has not propagated well enough to experimental biologists and computational biologists. Yet, without systematized information, prediction cannot easily work. On the other hand, computational biologists who use information from both experimentalists but predominantly from biocurators do not necessarily develop software that can be easily used by biocurators and experimental biologists to learn more about protein function and guide experiments. And so, although we find evidence of collaboration, we believe that much needs to improve in the quality of that collaboration between these groups. So we identify social challenges in this field, and perhaps they may stand in the way of the technical progress in protein function prediction. A brief note about the limitations of this work. This was a relatively small cohort. 82 scientists participated, and most of them gave opinions, and they had their own interpretation of the questions that we asked. We identified indeed a few minor problems with how they interpreted our question and what our actual intent was. And another issue in the survey was that, especially for experimental biologists, this was pretty time demanding. And by the end of the survey, we believe they may have got a little bit tired in how they responded to the survey. So this is our work. Thanks again to Rashika Ramola and Ida Friedberg, and I thank you very much.